What is autophagy? Autophagy is a process that is important for balancing sources of energy at critical times in development. It is also used in response to nutrient stress. The use of autophagy that is most commonly known of is used in cleaning the body out. It is known as an innate recycling system. It helps us get rid of faulty parts, stop cancerous growths, and use the molecules for energy or as building blocks for new parts. Autophagy can be used to remove misfolded or aggregated proteins, clear damaged organelles, as well as eliminate intracellular pathogens. Consequently, autophagy is quite often thought of as a survival mechanism. Although, if it is deregulated, it can cause cell death. The three main forms of autophagy are macro, micro, and chaperone-mediated autophagy.
The most common form of autophagy is macroautophagy, which is best known for its formation of a double membrane structure called an autophagosome. The process is induced by a stimulus, such as starvation, resulting in the formation of an autophagosome. Endosomal vesicles deliver lysosomal components, such as membrane proteins or enzymes, to the autophagosome. At this point, the autophagosomes are also called late or degradative autophagic vacuoles. Finally, autophagosomes fuse with either late endosomes, multivesicular bodies, or lysosomes. The autophagic body is delivered to the lumen to be degraded by enzymes such as lysosomal hydrolases. Microautophagy differs from macroautophagy due to the absence of the autophagosome, the intermediate vesicle that delivers cytosolic contents in the latter process. In microautophagy, cytosolic contents are directly taken up through invaginations in the lysosome membrane, leading to immediate degradation. Chaperone-mediated autophagy, also known as CMA, is another type of autophagy that differs from macroautophagy by the specific selection of cargo and delivery process to the lysosomal lumen for disposal. In CMA, chaperone and co-chaperone proteins recognize a specific pentapeptide sequence in all cytosolic proteins and form a chaperone substrate complex. The chaperone substrate complex is recognized by the lysosomal membrane receptor, resulting in their unfolding and degradation of the substrate protein via the lysosomal lumen. Macroautophagy and CMA have distinct processes but communicate with each other in the autophagy lysosomal degradation process. During certain conditions, one process can act as a backup mechanism when the other is defective. Our understanding of the relationship between these two processes is very limited. More research in the advantages and limitations of these two autophagic pathways is crucial to better develop effective therapeutic interventions in autophagy-associated human disorders.
Mediated degradation of damaged organelles, proteins, and lipids plays a critical role in the development of many age-related diseases. As a result, the process of autophagy serves as a novel pharmaceutical target in the development of therapies. Certain conditions can be improved with enhanced autophagy, such as neurodegradation and the pathogenesis of neurodegenerative diseases. Furthermore, the inhibition of autophagy also suggests beneficial health effects for conditions such as cancerous tumor growth. The full pharmacological potential of autophagy has yet to be uncovered.
A particular disease that relies on the interference of the autophagy pathway is Huntington's disease, an inherited neurodegenerative disorder. Huntington's is characterized by progressive motor disorders such as involuntary movements, cognitive deterioration ending in dementia, as well as psychiatric disturbances such as anxiety and addiction. Currently, therapies are evolving to upregulate autophagy where the Huntington protein is produced, as the mutated molecules rely on autophagosomes for clearance to reduce toxicity and degenerative effects.
To highlight the key components of this process, autophagy is used in times of stress to meet energy and nutrient demands, and also performs general housekeeping by eliminating damaged organelles and proteins. The main distinguishing characteristic of macroautophagy is that it forms a double membrane-bound vesicle, called an autophagosome, around the organelles and proteins to be degraded. Microautophagy occurs when the cytoplasmic constituents get directly consumed by the lysosome via invagination of the lysosomal membrane. Finally, chaperone-mediated autophagy involves chaperone proteins, which transport targeted proteins across the lysosomal membrane. With neurodegenerative diseases, cancer, and cardiac disorders being just some of the areas in which autophagy can play a role, we have yet to determine the true potential of therapies focusing on enhancing or inhibiting autophagy as necessary.